Section 4. Discordance and positive bias in germline and somatic variant classification. Section 4.1. ClinVar allows cross-comparison of classifications. In 2013, the NIH's National Center for Biotechnology Information expanded the concept of locus-specific variant databases and launched ClinVar as a public archive for clinical laboratories, researchers, expert panels, and others to archive their classifications of variants across all genes in the germline context, and more recently, in the somatic context. Sometimes ClinVar participants provide minimal or detailed evidence summaries with their submissions. A rating system has developed to reflect concordance and/or confidence in classifications, and the Food and Drug Administration, the FDA, now approves certain classifications found in ClinVar that are made by FDA-sanctioned expert committees. These gene or disease-specific committees vary in their reliance on evidence as opposed to expert opinion. ClinVar facilitates direct comparison of discordance among labs that submit to the archive. Section 4.2. Discordance and false positives in germline classifications. The 2015 ACMG-AMP Germline Variant Classification Guidelines provided additive algorithms and criteria for classifying pathogenic or likely pathogenic variants and benign or likely benign variants. Variants unable to be classified with these criteria or with unresolvable evidence conflicts remain VUSs until they are eventually clarified. Some evidence criteria carry recommendations for quantitative or semi-quantitative thresholds. More commonly, however, the evidence criteria are conceptual in nature. This is an inescapable result of clinical variant classification science being developed in the absence of large truth sets. In the absence of detailed and orthogonally validated truth sets, expert opinion is used to fill knowledge gaps. Unsurprisingly, studies comparing the application of the 2015 ACMG or AMP classification guidelines across different laboratories reveal discordance. Of particular importance is discordance that has a clinical impact, such as discrepancies between pathogenic or likely pathogenic variants and VUS. These discordances occur in a range of 1.5% to 10.8% among clinical laboratories. Examination of trends in discordance over time show that since the development and publication of the 2015 classification guidelines, overall clinical discordance has decreased to around 2.8%. This may also be a product of more communication between laboratories and integrated use of existing ClinVar classifications. Note, however, that concordance does not guarantee accuracy. In a 2018 study, researchers demonstrated that approximately 11.5% of genetic disorders already had enough variants cataloged as pathogenic or likely pathogenic in ClinVar to exceed the population prevalence of genetic disorders. Considering that ClinVar is an incomplete archive of genetic variants, the study's estimation of impact from false positive variant classifications represents a lower bound. The study also concluded that the false positive classification burden arose mostly from misclassified rare variants, rather than misclassified common variants. Section 4.3. Discordance and false positives in somatic classifications. In tumor genomic profiling tests, also known as somatic tests, multiple studies have shown extremely high discordance rates among different laboratories. When comparing final test results, it is difficult to determine which of the multiple complex laboratory steps lead to discordance. Although we know that current somatic variant classification technologies suffer from many classification false positives, where random acquired variants are often overinterpreted as driver variants. In one of the largest comparative studies published in 2019, 3 mimetic non-small cell lung cancer samples with 6 variants were sent to 101 different laboratories. 2 variants had above 85% concordant classifications, while 4 were highly discordant. Only 1/3 of the laboratories returned results correctly identifying all 6 expected variants.